Hello everyone, my goal with this video is to show you what to really look at and go over the context regarding Lab 1. These are the results that you'll have for Lab 2, in your case, for 2420. And this is going to be uploaded as a PDF on OneDrive. We have here exactly what you're supposed to be looking at. In the lab, we would have your TA, myself, Dr. Doyoungin, who would be able to go and show you some more interesting things that happen when we're looking at these bacteria. The goal of this is to show you what you're looking at here and how to use this document for your lab. I do have off to the side here your lab guides and handouts. So we are going to be going through lab guides and handouts, explaining exactly where things are. This is a template for the rest of the course as well, so that you have an idea of what goes where and when. The biggest thing that I can tell you is draw what you see. Describe what you see. If what you see can't be described for some reason, that's fine. Do the best you can. We're not going to be docking points from you guys simply because the pictures are off. We understand that the pictures can be off. I'm taking these with my cell phone. Not the best camera in the world. It's certainly not a professional grade camera. Now, if we look at our drop page, it's going to have what we're looking at, 2420 Lab 2 results. And then in this second box down here, this lower area, this is where I'm going to have all of the information you need. If you're looking through a microscope, it's going to have the microscope. Typically, it's going to be the Leica DM750, and any information that isn't in the pictures themselves. In our case, this has the coding for what bacteria we're looking at, what they're on. They're on nutrient agar and nutrient broth, and the temperature is the growth temperature here. Now, when we take a look at one of our plates, as you can see, this picture is a little blurry, but it is number 1, Pseudomonas Fluorescence, and there is some growth here. Now, let me pull up my drawing bit here on PowerPoint. Because this is going to give me a little bit of something to hopefully show you what you're supposed to be doing. Okay, so we're going to annotate up here. Now, this is supposed to be an isolation streak. Isolation streaks with Pseudomonas fluorescens are a bit difficult when we're only growing over 24 hours. Pseudomonas fluorescens, just based on what we grow them at, the temperature they grow at, and how long it takes 24 to 48 hours, typically close to the 48 hours. So if you take a look at how I did an isolation streak, it's here's a streak, here's a streak, here's a streak. Notice how I'm using the upper section of the plate. Then we have another streak here, here. I did not go back for bacteria. So after streaking, I disposed of my loop. Then down to, disposed of my loop. And then there, disposed of my loop. And if we let this grow a little longer, and you'll see with the Enterobacter in particular, you can get individual colonies towards the tailing edge. This is what's known as a T-streak. And it's called a T-streak because I'm going to show it over here. We divide our plate like so. And we have a streak here, then goes here, and then goes here. So we have quadrant 1, 2, 3. There's also things known as a quad streak, where we start in one corner, we go over, down, and over. The goal of this is to isolate individual colonies. For some of these, like the Pseudomonas, we needed to let it go a little longer. It didn't go all the way over into quadrant 3. It stayed in quadrant 2. Let's take a look at the next one. Now, this one you can really see where the streaks are. 3, 2, 1. And then right in here, and right down in here towards the tailing end, you do have individual colonies. Now, this is plate 2. It's Enterobacter aerogenes. Now, these are the Na plates. What you want to look at with these is you want to do your best to do size, shape, margin, surface, elevation, texture, and the like. It's not going to be easy, and you do the best you can with the pictures that we were given. The hard part is a lot of these you'll be able to pick up the plate, turn the plate, look at the plate, that type of thing that you really can't do on a picture. Do the best you can with the tools that you are given. We are not going to dock points for you simply because you can't find something. The best way to describe it is with the Pseudomonas fluorescence. You're getting some growth here, but not a lot. Do the best. Now, if we take a look here at the Cocoria, same thing, 3 here, 2, 1. There would be growth over here. Later on in the semester, I'll show you a very good isolation streak. It'll probably be a quad streak. That'll show you exactly what you're looking for, but we don't have those growing at the moment. And this is Bacillus megaterium, and as you can see, Bacillus tends to take over a plate. This was not after 24 hours, this was after 18. So after 18 hours, it just takes over everything. On your lab guides and worksheets, fill in the table as best as you can using the guides inside your lab manual. Same thing with the slants. Here are the slants for number 1, 2, 3, and 4. Do the best you can to fill in texture, optical properties, margin, and color. Optical properties are not possible. The reason they're not possible is because you need to have a light behind the plate or the slant, which you can't do at home. And with our pictures, you really don't see much even when we put it on a plate reader. So you do not have to fill in optical properties. We will not take off points for that. Here's your broths. There's number 1, 2, 3, and 4. Again, describing the growth patterns that you find in your lab manual. Now, as I said, it's going to be a touch difficult with pictures, but in the lab, if you want to come in the lab, we could go over with you how to do a T-streak, how to do a quadrant streak, and using bacteria, what things look like. Another interesting thing regarding the Enterobacter and the Pseudomonas. Both of them, when they get really strong growths, they change from this clear color that you see on both of them. They change the auger. The Pseudomonas fluorescence is known as Pseudomonas fluorescence because it has a neon yellow color to it. Well, a neon yellowish green color, depending on the strain. I believe ours is more on the green side. It's not really highlight green, but it's more like a lime green is what it turns the auger and the individual colonies. Whereas the Enterobacter erogenes is sort of a pink. Because of that, that would be a way to describe it. We look at Cocoria rosea. The individual colonies themselves are pink, and that's different than the colonial mass. The colonial mass of Enterobacter erogenes is a slight pink color, but as you can see, the individual colonies themselves are clear. Whereas Cocoria rosea, the individual colonies are a pink color. This is called chromogenesis. And chromogenesis is just a changing of the colors. Some of them really make it obvious what they are. Cocoria rosea is pink. Cocoria rhizophilia is a yellow color. Your Sarcina aurantiaca is orange. And the chromogenesis is a big way of determining what you're looking at from an outside perspective.